the Deep Sea podcast, Pressurised, a short, punchy version of our main feed that gets right to the scientific point. If you like what you hear and you'd like to hear the full episode, you can find it in the same feed. And now, to get right to the point. So I had a little bit of history with cold water or deep sea corals many years ago, much younger man. I was part of the Coral Fish Project, which was an EU 7th framework project that wanted to look at the association between deep water, cold water, reef-forming corals, mainly the Lophelia, which I think has changed its name now, and Madrepora, lattice and reef-forming corals down there, so the stony corals. And the idea was that they are enriching fish communities, basically, by providing a habitat. So the project came to an end and the results were supportive. The sort of research bed that it created continued to tick over. We now know that certain species of skate and small sharks deliberately place their egg cases in the coral reefs, and actually the shape of the egg cases seem to be evolved to tangle and hold onto these coral lattices. And they're also sort of habitat engineers. They change the habitat where they grow. They change the current flow and they sort of clog with sediment, and they've got all these associated species. So it is like a building block of a habitat. And yeah, that was an important piece of work for me. It's even immortalized in my tattoo sleeve. It ends with a cold water coral reef based on that work. A few listeners on the Discord were asking for an episode about deep sea corals, and around these parts there is no one else really to speak to other than Tracey, who is a bit of a local legend. She is a coral expert at NIWA, but came into it through a sort of fisheries and fragile habitat advocate and researcher. So champion of the vulnerable deep sea habitats, fishes and coral alike, and just a really charming person who actually really supports the scientific community here. She has nurtured and mentored the next generation of scientists here as well. So we're very lucky to be joined by Tracey for a chat about deep sea corals. I'm lucky enough to be joined by Tracey, who often goes by Di. I think I've always known you as Di.
Yes, kia ora, Thom, and great to be here. Great to chat about deep sea corals, actually, because you keep thinking, everyone knows about deep sea corals now. We've been looking at them since 2008 in this region, and actually they don't. They're just familiar with the Great Barrier Reef corals. So thanks for the chance.
Yeah, they have a sort of forgotten status, even though we've been going on about them for so long. I wanted to start with a bit of a foundation, because a lot of people are sort of aware of corals, but not their structure as an animal. So can you talk through just what a coral is, how this colonial organism sort of functions?
That's right. They are animals, and the little polyp that lives on the animal is a bit like an anemone. So each polyp is an individual animal, but they have an exoskeleton. So they build in a tree-like way, often, a skeleton with branches. And it's on those branches that the polyp lives, often embedded in the skeleton, and so you can't see it, but then it protrudes with the tentacles when it's feeding and breathing. Yeah, they're animals like anemones and jellyfish are in the same group of Cnidaria. The skeletons are formed often using calcium carbonate, which they get from diet, but also from just the natural minerals in the ocean.
Is there a sort of continuous tissue? Are the polyps connected to each other?
In some, yes, there is. The senenchyme, or some people call it the cenosac. That's the tissue that is covering the skeleton and connecting the polyps as well. There's not loads known about how that connection exists, but if the senenchyme disappears, the coral's not doing very well as an animal. So yeah, within each polyp, there's this pink tissue, for example, on some corals that connects along the branches to the next polyp. That's external. Internal, there's also stuff happening as well.
Is it one sort of cloned organism? Is a single colony sort of genetically uniform?
Yes, we think so. That's correct, yeah. And there are male and female colonies also.
Oh, right, at the colony level rather than the polyp level. Oh, that's interesting.
Yes, that's what we believe, certainly for the scleractinian stony coral. So one clump will be a male clump and the other one will be female. So the female will produce the oocytes and then the spermatophores come from the adjacent clump. Yeah, but that kind of connection is quite tricky.
Yeah, I bet.
And then some brood the animal inside the mouth of the polyp. So you've got that going on as well.
They're quite tricky in their simplicity. So there's just one entrance / exit. So they eat and they expel eggs and sperm and, by the sounds of it, expel even juveniles just through this single opening in the middle.
That's right, yeah, the cavity. The ones that brood the larvae, that would be in what we call the mouth of the polyp. So that's just sitting in there amongst all the rest of the organs of the coral. And then, it just pops them out when they're ready.
Oh.
And off they swim, which is amazing.
Yeah, and they must have to halt all digestion. They must have to halt a lot of other processes in order to do that.
Focus on the breeding. Yeah, that's right.
Yeah, single cavity. Is a single colony functionally immortal? Or because it's a clone, is it sort of building up genetic damage? Is it important to have that sexual reproduction as well to get some variety in there?
You would have to have that. You also would have to have the animals being far enough apart so that the genetic material is more variable.
Yeah.
I'm just going to butt in here and say I'm not a taxonomist and I'm not a geneticist. I'm not a molecular person. My career began with us not even knowing about these animals. We didn't know where they were. We didn't know what they were.
Started on day one.
Yeah, it was, the focus was on single species stock assessment when my career began. So we were looking at how many fish are in the ocean, how many orange ruffy are on these seamounts or in this area, and then focusing more on the bycatch, what we were also getting in the trawl, the other fishes, as well as the invertebrates, led us to do a lot more work on invertebrate species in our region. And that's when we started with the coral research, the spatial distribution knowledge. Where are the hotspots?
This came from a sort of fisheries element because it's important for the fish. It is important habitat. These are not just animals in their own right. They shape the habitat around them.
Exactly. Yeah. So, they are very important. You'll hear that word, importance of habitat, throughout. Anyone who talks about corals will mention habitat and importance and associates what lives on the corals, what species use them for shelter and refuge, or what species really rely on them as well, for platforms to get into the water column to feed. So all that kind of information and knowledge has grown for me since we did the fisheries work. Of course, there were taxonomists in the museums in New Zealand looking at the corals around our coast, and that work was going on. And when they put the cable between the North and South Island and lifted it for maintenance, they found corals on that cable. So there was work going on in the early days, in the 60s and 70s, but that was more coastal. The deep sea and that knowledge really just started with the growth of the deep sea fisheries and knowledge of what was on the seamounts.
Yeah, things were just coming up in the bycatch that never seen before. And
exactly.
Some of the images from those early days, huge corals coming up with the trawls.
And those huge corals still come up with the trawls. Because, people are always going to new areas to look for more fisheries or more stocks, and they're bringing up corals still. We're continuously doing work on protected corals for the Department of Conservation. We're monitoring bycatch on fishing vessels, and we're still seeing those really large corals coming up from the deep. For example, the bubblegum coral, that charismatic Paragorgia, that's still being brought up in trawls as we speak.
And what would the age of a colony like that be? I know they're incredibly slow growing.
Well, the bubblegum corals don't seem to be as slow growing as some of the other coral groups. So we'll be getting black corals living to over 2,000 years. But the bubblegums seem to live, to be honest, we've only radiocarbon dated one specimen, 300 to 500 years. So yes, old, but not 2,000 years. They seem to have quite a fast growth rate. It might be because they're not calcium carbonate structures. They're more gorgonin, chitinous.
If they're well fed, they can lay it down pretty quick.
Yeah.
Should we talk about the two main structural groups? How they make their skeletons sort of impacts the flavor of the coral quite a lot. If they're slow growing and hard or sort of proteinaceous and a bit flexible.
Right. So there's the scleractinian stony corals, that protected reef-like coral that we have in our region that's made of calcium carbonate. And so they are very slow growing. It could take about 2,000 years for 1 meter of that colony to grow. So that's one key group, the calcium carbonate structures. Scleractinian stony corals. Then there's the bamboo corals, which are a mixture of calcium carbonate and gorgonin. And they are also very slow growing, but not as slow as the stony corals. And then we look at the black corals. that's made up of a different material, kind of more chitinous. They can be quite fast growing in the Fiordland region and live to about 300 years. But in the deep sea, they're very slow growing, and we've aged them to over 2,000 years as well.
Even the very youngest of the ones you mentioned are older than our ability to impact them. We've only been able to bump into these things for the last 100 or so years at the very most. You know, the Challenger, maybe it was a few pretty specialist expeditions, and it's only really now last 30 years, maybe last 40 if you're pushing it, where we've really been impacting these very, very slow growing animals. So they've not really had a chance to rally or adjust.
No, not at all. We've carried out recovery experiments. We've been out to the seamounts and looked at where there's been damage to the colonies, and then we've looked for signs of recovery. Are the corals growing back along those tracks that have been heavily trawled? And we're not really seeing any signs of the growth. What seems to happen with the stony corals, which my colleague has been studying, Jennifer Beaumont, the reproduction work, was that we've seen the larvae brooding inside one of the species, Goniocorella, and we've seen those larvae come out and settle really quickly and start laying down a skeleton to get some kind of base, get some hold on something hard and on a suitable substrate. And then the growth would slow. So it's initially, let's get settled and start growing a skeleton so that we're safe and we can start growing. That seems to be a reasonably fast phase of growth, that initial one, and you see that in a lot of animals, but then it slows.
Let's talk about the corals as habitat engineers. So say an empty seamount or a seamount that's been impacted by industry. What changes are coral colonization sort of offering? How do they change the habitat?
So a lot of the seamounts we've studied on the Chatham Rise, a few are protected. And so we can look at those pristine environments and see how the corals behaving there and see what they provide as far as habitat for many other invertebrates, not just the live matrix, but the dead matrix as well. So we can observe side by side an impacted seamount, what importance the corals have. They don't just provide habitat for other invertebrates, they're also important for other fisheries. You know, the fish either feed on the animals that feed on the corals or feed on the animals within the corals, or they use that coral matrix. They're using that for refuge and shelter as well. So quite an important role.
It's beyond the immediate habitat. They engineer and they influence quite a halo around themselves because they change fluid dynamics as well, and they bring more nutrients into the area really by changing the fluid flow and the productivity in the area. So they seem to enrich quite far beyond their colonies.
I mean, that's been hugely studied in the Great Barrier Reef area, where you're seeing a coral and the coral dinoflagellate symbiotic animals. You're seeing them maintaining at an individual level that kind of environment. We really don't know how much that is happening in the deep sea. That's kind of a huge area, but you're right. Those corals, that halo of corals that are draped over the flanks of the seamounts, are also changing the whole environment at a local level. Upwellings that occur with the oceanography around seamounts is a huge thing, and that's where you'll get a lot more feed, but that's beneficial to the corals and the animals that live within the corals.
I realise we've missed something quite fundamental when we're talking about deep corals versus shallower, which is the lack of the symbiotic alga.
That's right. All of the corals that we look at in the deep sea, they are azooxanthellate. They don't have these dinoflagellates, so they do their own breathing and feeding. Whereas in the shallows and Great Barrier Reef, they're helping the corals survive by doing the breathing and providing the food, and also maintaining the ocean environment around the corals, so keeping that pH at a level that the corals like. But they're battling climate change at the same time, so that's where you get the bleaching events, and those dinoflagellates die. They're the ones that bring the colour to the coral, and so you just see bleaching and pale corals. So we're not seeing that in New Zealand because our corals are doing their own breathing and maintaining their environment. But they still get stressed because the ocean chemistry is changing, as well as the temperature in our oceans. And so when that changes, you can't form the skeletons. So they are suffering in a different way, but and just as serious as the shallow corals.
Maybe less visually striking.
Yes.
The bleaching events are quite immediate and quite sort of
That's
sudden,
right.
but is it more subtle with the deep ones? It's hard to see when they're stressed.
It is. Our deep ones are very colourful when they're healthy, actually. They're very pink and red and yellow and purple. They're pretty stunning. So you would see changes because of temperature, and that senenchyme that we talked about earlier, the tissue that surrounds it, that pink tissue would disappear. But yeah, be more subtle. We're seeing those massive bleaching events are very dramatic, seen from space, and yeah.
Yeah, and like happening over a week.
That's right. Yeah.
It's going to be more gradual, and it's hard to right the ship in the deep sea. And we're seeing more and more, well, maybe it's always been there and we just haven't recorded it, but we're seeing more and more sort of high temperatures rolling through the deep sea. And because of the crazy current regimes where it's often detached from the surface could be fine, but a pulse of warm water has gone through the deep sea and stressed a load of things in a way they haven't done before.
That's right. We're seeing those hotspots in New Zealand already. So yeah, it is serious for the shallow and the deep sea corals.
When you're growing over hundreds of years, they can't dodge out the way like fish finding a new optimum.
Well, I mean, you mentioned earlier in the interview this transplanting at sea that's going on, and it's not just going on in shallow water corals, but in the deep sea as well. They're taking up colonies and growing them in an aquarium and then settling them back into those deep sea environments.
I did not know that. I know you in NIWA, you've kept them alive in the labs there.
Yeah, we maintained them in our aquarium, and they lived for well over a year in a cold, dark environment that mimicked their natural environment. And the first work we did was on ocean acidification impacts, so looking at changes in pH and how they would affect the corals. So that was one experiment we did, and over a long period we certainly saw impacts, dead polyps and the senenchyme, that pink tissue, disappearing from the skeletons. And then the second study was looking at sedimentation. So how did elevated levels of sedimentation impact the corals? And again, we saw, pretty robust life with the coral results. The corals were doing okay for a short period of time. But as the sedimentation accumulated over time, we could see stress on the corals again. The tentacles weren't able to flick out the sediment adequately and they would die, or that senenchyme tissue would just be disappearing over time.
They can cope to a point, but then it's quite an abrupt point where it's just too much and the colony is struggling.
I mean, they often live in quite a high sediment environment. Some corals live on seamounts where it's hard bottom, rocks, and not much sand or sediment. And then other corals will live on small stony structures on flat sediment. So to a certain extent, they can maintain themselves and stay alive in those environments. But when you add additional stress, that's when they struggle. So yeah, they're not all found on hard rocky outcrops or on the sides of canyons. They're also found in soft sediment. We have the stony corals, the cup form, not the reef form I've talked about a lot. Those cup corals can be on hard structures like other corals or on rocks. You have the reef-forming corals that attach to rocky substrate and seamounts and drape themselves all the way down the flanks of seamounts. But you also have the cup corals, and they can live on hard substrate or just in sediment. They have a different structure.
Are they the ones that tend to be solitary? They look like an armored sea anemone. They make these cool tooth-like plugs.
Yeah, solitary cup corals, yeah.
I know it's present in a lot of the stony corals, but because they're so much bigger, you really get to see how beautiful that structure is, like how much the flesh of the animal is quite small, and there's this beautiful sort of ridges to the cup itself.
Yeah, the septa.
Is that what it's called?
Yeah, and they have these different patterns. Yeah, they're amazing. And then the column can be long and elongate or flat and like a bowl. And yeah, they're really beautiful. You're right.
Is there any sharing of resources through the connecting membrane, or is each polyp on its own?
Yeah, we talked about that a little bit earlier. I think there's sharing of resources, but I have no real understanding of it. They seem to be able to survive on their own when we've taken the samples back, and they haven't seemed to be dependent on the polyp next door. But there must be connections. There's often hollow tubes between some of the corals, not all, but that stony branching form, the branches are hollow.
Connection was sort of running through them. I always thought of like the nerves of a tooth. There's a little core of flesh that goes through.
But the other corals, the bubblegum and the black corals, that skeleton's solid, and you can't really see that they would share. It's a hard structure. So perhaps it's the external tissue that shares between polyps and not the internal.
Oh, that's interesting.
Yeah, it's tricky. I mean, if you cut open a Scleractinian stony coral branch, you'll see those tubes, but you'll also see divisions, like it stops. It's like a wall. And so you think, what's going on there?
What are the sort of common commensal organisms? Who are the friends of the corals? And I suppose there's going to be predators and things that graze on them as well.
Yes, the echinoids graze on them, so I'm not a fan of them. So those deep sea urchins, they'll move across the seamount and start nibbling on the coral polyp. The worms that we've already mentioned about, the polychaetes, the marine polychaetes, they seem to be more commensal, and it seems to be okay. The galatheids, the little squat lobsters, use them as a platform, but they're not really, in a symbiotic or commensal relationship. They just use it as a structure to get into the water column to feed. Lots of ophiuroids are found, the little sea stars are found on the corals. That's probably it for the kind of the commensal animals. If you're looking at those Gorgonian octocorals I mentioned earlier, they seem to have these ophiuroids that are always on them, and they just seem to live in more of a commensal way. They're not being eaten. They're just, those snake stars are just twisted all around the branches, and
I've seen
really that. Abundant. Yeah, so that's on those what used to be called Plexauridae. They've changed the family name now. So they have them. And black corals in the Fiordland region, it's been observed that they have that same snake star that will live in association with those huge black corals forever. They've been studied by scientists who have been in the Fiordland area since the 70s, and they see that same colony with those same snake stars. They're all happily kind of living together.
Oh, it's a long-term one. And then I know a lot of fish, particularly the elasmos, there's some skates and some small dogfish that seem to really target the coral lattice to lay their eggs, and the egg cases actually seem to be structured so that they coil and wrap around the coral and get secured that way.
That's exactly right. In fact, my colleague Britt Panucci, whom you've interviewed, I believe, always says, look out for the skate cases on the coral matrix. Yep, that's right. So very important structures. There's always a seamount we go to on the Chatham Rise, and we see these two giant lepidians, these huge fish. They're always in the shot, and you think, what are they doing there? Are they just sheltering and using it for refuge and passing through? But they're large, and they always seem to be hanging out there. So they must love that environment. They must be feeding on something there. But smaller fish, we've seen little morid cods sheltering in their branches.
Yeah, cusk eels too. Cusk eels seem to have a home coral sometimes.
That's right, yeah.
Did you want to talk a little bit about sort of career progression? Because you were on the front line of getting to see, and I know you've done a lot of work for improving the offshore environment, particularly for women in science. That was a listener request. You've seen the industry change, and you've sort of driven a change in that area. Because even the 20 years I've been going to sea, the attitude has really changed. We still get fisheries observers writing in saying, like, I keep finding garlic under my pillow because they think I'm a witch. And
yeah, well, that's pretty sad, isn't it? You think things have changed. I started in the 70s, so it was a long time ago. And so I was 4 years working in the Fisheries Research Division. It was called the Marine Department, and then it became Ministry of Agriculture and Fisheries. And women had just started to go to sea vessel, the James Cook. I think the first woman, Anne Brunt, who went on that vessel, the director had signed off their voyage program without reading the names. And that's how we got a woman onto that trip. And then for those 4 years that I was working, there were always 2 or 3 women going on that research boat. And that was a pretty good, equitable experience, to be honest. Young technician that I was, learning about the job and learning how it was to be on research vessels. It was all a good experience. You often come to awareness about equity when you're a little bit older, and you go, hold on a minute. they're doing the same job as me. And I've been doing this for years and they're paid more than me. So that kind of awareness came when I got back from overseas, when the exclusive economic zone had just been declared. The EEZ suddenly was 200 miles of waters to research.
Well, so you were part of that transition, because it's so galvanized now, the EEZ. But it's not, it's a really new thing. Like there was a time when it was the 12 mile.
We didn't have a research vessel that could really perform in that whole region. You know, it was huge. And so we relied on initially foreign vessels that were in the region, on Japanese vessels. We were doing research. Russian, German, the Wesermunde was there for 2 years, assessing the size of our resources. But yeah, I was often the only woman on those trips, and kind of relied on the support of my male colleagues to make sure everything went okay. Yeah, because it was very unusual for women to go on those voyages. And then we spent 10 years going on the New Zealand research commercial vessels before we got our research vessel, the Tangaroa. So we used commercial boats to do research for quota or research for money surveys on all the new stocks that were being discovered, orange ruffy, Oreo species. This was mainly a deep sea focus. If I was leading the voyage, I always made sure I had women on my team. You know, we're part of 50% of the population, and of course we bring value to the job, and we do things slightly differently sometimes and add a huge dimension. And sometimes that was hard to get the women on the trips, and sometimes it was a struggle in those environments because you did have sexism and pornography in the workplace.
Yeah. Even during my career, that's really changed. Like there's just the atmosphere offshore has changed.
Yes. Yeah. Well, good. So I think, that was a time of making sure people heard women, and we showed that we were capable of leading voyages, and several women that I worked with led voyages in the 70s and 80s. One of those women would say maybe it was a bit easier then. We had equal employment opportunities programs active, and so there was a big focus on equity and fairness and women's rights. And then, of course, so that was a bit of a stick, so the men had to comply with what the government was saying. EEO programs were strong. They had a lot of clout. But then over time, sometimes it was hard for women who had families to keep going to sea. So I didn't have children. And so often, even in the 90s, I was still often the only woman at sea. But yeah, it was, as you say, the environment was loads better. We'd kind of addressed the issues of no pornography in the workplace. We had rules, and sexist behavior wasn't as common. And now we seem to have that, a lot of women are going to sea and they're leading voyages, and they sometimes have an all-woman team, but that still gets questioned, which I find really intriguing. Gosh, how would it be on that voyage? It's all women. Who would do the lifting, for example?
What if you get a really big rock?
So we still get that, that bias, and it's a bit more underground, you could say, that unconscious bias. But yeah, throughout my whole career, I've tried to support women, because it has been a very non-traditional role. And, 2 women at sea was much easier than 1, or you'd quickly get to know the steward, who might be a woman, and you knew that you had allies on board. So that really helped if there were any issues or any kind of harassment issues. And then as I pass on the mantle of the coral research, I'm probably a bit biased myself, and made sure that I've passed it on to a lot of, great women researchers. So there's women who are coming through now who can pick up most of this work. Here's a shout out to women researchers. Thanks for the question.
Well, yeah, I want to talk about that, because in my time, I've seen a huge change. And I just think the more different your team is, the better you work. Yeah. You can have the smartest, most capable people, but if they're all the same type of smart and capable, they only have the same idea. Yeah. I really like sort of mixed, diverse teams, because someone at the back of the room will have this totally different experience or this different way of thinking.
Exactly.
Thanks so much for your time today.
Thanks, Thom. Nō reira, tēnā tātou katoa.
Oh, nice. I'm still brushing up my te reo. I need to get more confident with it. Even if I'm doing it wrong, I just need to go for it.
Yeah, get your greetings sorted. I presented this really nice one at my retirement party, when the old net is cast aside, the new net goes fishing. And that's kind of about the old net has one way to do things, but the new technology can do it slightly differently. And that's how I see passing on my work to these great mid-career women who add this great new value. So it is kapu te ruha, tahau te rangatahi. When the old net is cast aside, the new net goes fishing. Kia ora.
And that was a pressurized version of one of our longer episodes. If you enjoyed that and you would like to hear the full-length episode, just match the episode numbers and you'll be able to find the full-length version in the feed. Thanks for listening. We'll deep see you next time, and I abyss you already.